Controlled traps have been somewhat effective at keeping feral hogs in check. There's also been extensive research into developing a legal poison, but nothing has been approved here in the States.
The biologist made her first cut, expecting the usual: acorns, roots, corn kernels from the fields nearby. Feral hogs eat almost anything, and this 220-pound animal from eastern North Carolina had been raiding farmland for weeks before wildlife officers caught up with it. What poured out onto the stainless steel tray was not the usual: bright plastic, shards of water bottle, thin strips of shopping bag film, small hard fragments that looked like packaging. Dr. Elena Vargas, a wildlife biologist with North Carolina State University, had seen plastic inside hogs before. The animals raid dumps and roadside trash. That part was not the problem. The problem was what the plastic looked like now that it was out of the animal. The edges were wrong. They were not chewed. They were not torn. They looked softened, as if something had been slowly working on them from the inside. She called her colleague, Dr. Raj Patel, over to the table. They stood there in silence for a long moment, holding pieces of a water bottle that appeared to have been chemically altered inside the stomach of a wild pig in the North Carolina coastal plain. Nothing in either scientist's training explained what they were looking at. Feral hogs are the single worst invasive species most farmers in North Carolina have ever dealt with. The animals are now present in 85% of the state's counties. They tear up wetlands, destroy the ground nests of native birds, and kill the fawns of white-tailed deer. In the eastern part of the state, where peanuts, corn, and soybeans grow, a single sounder of hogs can wipe out thousands of dollars of crops in one night. They damage drainage ditches, tear up levees, and undo years of conservation work in a single week. Across the country, the damage adds up to somewhere between $1.5 and $2.5 billion every year, and the population keeps growing because a single sow can produce 2 litters a year with up to a dozen piglets each. That was the problem Vargas's team was trying to help solve. Their diet study was supposed to be practical: find out what the hogs were targeting on the landscape, then help managers place bait more effectively and focus trapping in the right places. Nothing about the project was designed to look at plastic, but the plastic kept showing up in hogs from certain counties, and after a few weeks of dissections, the pattern was too consistent to ignore. Most biologists would have written it up as incidental ingestion, a note in the report. Hogs eat trash. Everyone knows that. Move on. Vargas and Patel did not move on. They brought in a third scientist, Dr. Priya Sharma, a microbiologist at the same university who studied environmental microbes. Together they made a decision that most wildlife departments would not have signed off on. Instead of treating the plastic as a curiosity, they were going to treat it as a possible biological signal. They would sequence the microbial communities living inside these animals. They would run controlled experiments to see whether anything inside a feral hog was actually capable of interacting with plastic on a chemical level. That decision cost them. It cost them time. It cost them money the department had not budgeted for. And for months, it cost them results. The first sequencing runs came back with almost nothing. Common gut bacteria. Nothing that stood out. When they tried to culture microbes from the samples in the lab, most of the organisms died or refused to grow. When they tested those that survived against strips of plastic in controlled conditions, nothing happened. A senior researcher at a wildlife conference told Vargas politely that she was probably just documenting the fact that pigs eat garbage. A grant reviewer wrote that the work looked like a distraction from real management questions. A graduate student who had been excited about the project quietly asked to be moved to a different study. There were nights when Vargas sat in her office and wondered if she had built an expensive dead end into her own program. The plastic could simply be passing through. The softened edges could be nothing more than mechanical wear inside a stomach full of grit and grinding food. Without hard evidence that something biological was happening, the side project was going to sink the credibility of everything else her lab was doing. That was where the story could have ended. Another interesting anomaly. Filed away. Forgotten. It did not end there. The break came on a Tuesday afternoon in the microbiology wing. Sharma's team had switched to a newer sequencing approach that could read much longer stretches of microbial DNA at a time. The older method chopped the genetic material into small pieces that were often hard to reassemble. The newer method could capture whole gene clusters intact. When those results printed out, one group of samples pulled the whole team into the same room. Inside the guts of several hogs from the eastern counties, the sequencer had found bacteria carrying genes that coded for enzymes very similar to the plastic-degrading proteins first identified at a Japanese recycling plant and later found in certain ocean microbes. These were not the exact same species, but the working parts, the genes that actually did the job of breaking apart the chemical bonds inside plastic, were present. In some animals, those genes appeared in multiple copies, spread across different bacterial groups that seemed to be living together as a functional community. They ran the next round of experiments with much tighter controls, anaerobic conditions matching the inside of a real hog stomach, small squares of PET plastic, the same material used in most water bottles, microbial extracts taken directly from the hog samples. After several days, they weighed the squares again. The most active samples had lost between 4 and 7% of their original mass. Fourier transform infrared spectroscopy, a technique that can detect changes in the molecular structure of a material, confirmed what the mass loss suggested. The plastic was not just being ground down. Chemical bonds were breaking. Something living inside these animals was actually eating parts of the plastic they were swallowing. And the pattern held. Hogs taken from cleaner, less disturbed areas showed almost none of these genes. Their microbial communities produced no measurable change in the same tests. The adaptation was tied to the environment. Somewhere along the way, in the wetlands and drainage ditches and roadside dumps of the coastal plain, these animals had either picked up microbes from the soil and water or had amplified populations of bacteria already living inside them at very low levels. Either way, what they had inside them now was a functioning microbial community that scientists had never documented in a wild mammal in the United States. If this is the kind of wildlife story you actually want to hear more of, tap the subscribe button and turn notifications on. It costs you nothing, and it tells the algorithm to keep sending stories like this one to the people who care about what is really happening in American wildlife. This finding does not make feral hogs any less of a problem. That has to be said clearly. These animals are still destroying farms across North Carolina. They are still pressuring native wildlife. They are still going to require active trapping and hunting for the foreseeable future. Nothing in this research changes any of that. What it changes is our understanding of how far wildlife can go when it lives inside a landscape we have completely reshaped. Plastic pollution is now so widespread that it reaches even the animals we consider pests. And in the process, some of those animals appear to be building biological capabilities that were never part of their original ecology. The same feral hogs costing farmers millions of dollars a year in eastern North Carolina may also be carrying microbial communities that could one day inform new approaches to breaking down plastic waste in controlled industrial settings. That possibility is still years away. Nobody is suggesting that plastic-eating pigs are a solution to anything. Moving from a discovery inside a wild animal to a real industrial process would require enormous additional research, safety testing, and engineering, and most attempts to scale up microbial discoveries never make it out of the lab. But the first step, proving that this capability exists in a real, free-ranging mammal, has now been taken. The other thing this discovery does is quietly rearrange the way researchers should be thinking about invasive species. Plastic pollution and invasive wildlife are usually studied as two separate crises. Different journals, different funding, different departments. Here they overlap inside the same animals. The hogs did not create the plastic problem, and they are certainly not solving it. But they are living in the middle of it, and a small piece of what they are eating is being changed by what is living inside them. For the farmers in eastern North Carolina, the practical picture has not changed. The traps still need to be set, the permits still need to be filed, the nightly damage still needs to be reported, but the animals moving across those fields are more complicated than they were a few years ago. They are not just destructive pests. They are also, at least in some cases, walking laboratories, carrying microbial communities that our best science is only beginning to understand. So I want to leave you with a question. If two of the biggest environmental problems in the country, plastic waste and invasive species, are already interacting inside real animals in a way nobody predicted, How many other adaptations like this are happening right now in wildlife we rarely study? What else is going on inside animals that we assume we already understand? Drop your honest answer in the comments. I read them all, and the best ones often decide what we investigate next. If you want the next story the moment it goes up, subscribe and turn on notifications. Thank you for watching the whole way through, and I will see you in the next one.